Okay, so today's topic is transcription. Transcription, if we talk about the central dogma, there are two parts of central dogma. Number one is the transcription, and the other one is the translation. So in transcription, the DNA is transcribed into RNA, and in translation, the RNA is translated into the proteins. So in the central dogma, the direction is from DNA, and then DNA to RNA, and then RNA to protein. And in fact, that protein that shows the phenotype. So first step would be the transcription, and the second step would be the translation in central dogma. Now, here is the gene, and we know that DNA is double-stranded structure, and one of its strands 3' to 5', and normally we read DNA from 5' to 3'. So that will become the template. Now that template is going to be transcribed. That is the basically RNA transcribed as RNA, and now the difference between DNA and the RNA, we know that every T it will be transcribed as U, while rest of the nucleotides they will remain same. So you see the template is 3' to 5', while the mRNA is 5' to 3'. So each triplet we know it codes for one amino acid, and there are 20 or more amino acids. And then next comes the translation, where, for example, the first triplet here is the AUG, or if we talk about the DNA, it would be A3G, and that's in code for the methionine amino acid. And likewise, it will continue until the polypeptide chain is completed. This is a brief of central dogma. Now, DNA is associated with the chromosomes, we know, and from chromosomes, this DNA would be unwound in a simple form and in a linear form, so that the cellular machinery can read it and transcribe it into the RNA. And it is nucleus is present in the nucleus of the cell in eukaryotes. Chromosome, and chromosome definitely it is formed of the DNA. Now the first step is synthesis of the RNA from the DNA. That's called the transcription, and this occurs inside the nucleus. While the second step, as I earlier mentioned, is the migration of that RNA into the cytoplasm, where with the help of the ribosome, it will be translated into the protein. Protein synthesis occurs with the association of the ribosomes. So basically, ribosomes are the body which will help to produce the protein. Now, transcription by definition, we know this is the basically to transcribe DNA into the RNA, where each T is converted into U. So this is RNA is basically, we know it is a single-stranded molecule. First time it was described in 1961 by Francois Jacob and Jacques Mohammed, this mechanism of the existing of the RNA, and then later on it was discovered how it is transcribed and for that they use the triplet binding assay. In 1964, after 3 years of the discovery of the RNA, Sunderberg and Philip Ladder, they developed triplet binding assay to exactly describe the each triplet which is encoding for the respective amino acid. So there is specific sequence for assignment of each triplet codon, and the triplet RNA sequence is acts like codon. So 3 base pair together, they are a codon. It's not 1, it's not 2, it's not 4. It's exactly 3 which are encoding for the codon. Now triplet codon attracts the transfer RNA and molecular containing complementary sequence. Each transfer RNA can carry specific amino acid. So in cellular machinery, other than the messenger RNA, there is transfer RNA which is carrying the amino acid, and this is definitely a triplet just like present in the mRNA, but it would be complement to the mRNA triplet. And this is called the anticodon, that's triplet sequence in transfer RNA. Anticodon because it's anti-complement to the actual codon on the mRNA. Now here is the example. For example, this is synthetic RNA that is carrying triple U, and then the transfer RNA which is carrying triple A, and it is triple A is encode for the phenylalanine, and that attached with this one. And we know there are 2 subunits of the ribosome, and each one carrying of them. The one is carrying this transfer RNA which is bringing the phenylalanine in this case, and when the code comes triple U, it will pose, the ribosome will pose the phenylalanine, and it will add to the polypeptide chain. So here is the joining together, and the phenylalanine will be added to the polypeptide chain. So this is basically, you see the anticodon which is carrying, and that is transfer RNA which is carrying the amino acid, and triplet codon against that is basically messenger RNA, a component of messenger RNA consisting of 3 amino acids. So consisting of 3 nucleotides. Now RNA triplet transfer RNA ribosome complex. So there are 3 things involved: RNA triplet, basically the messenger RNA, and the transfer RNA, and the third is the ribosome machinery which is actually manufacturing the polypeptide chain. So these are the specific trinucleotides which represent respective amino acid. AAA or AA-G, this is lysine, and likewise that to the end, there are 2 codons. One is triple U, other is double UC, and that is phenylalanine. So among all these codons, if you see the first 2 letters of each codon for any given amino acid, they are same. For example, if we take the first one, so there is AA at the start, and the difference is in the 3rd letter. Here in the first codon, this is the capital A, while in the second it is the G. So that's the difference. So that means the first 2 letters of the codon, they are very important for determining the amino acid. So that's why any mutation in these first 2 letters, they are actually going to change the amino acid. While the 3rd letter, sometimes it can change the amino acid, and sometimes it does not change. It does not have any effect on the amino acid in the polypeptide chain. Now there is a dictionary of genetic coding, and there are 61 triplet codons is assigned to the amino acids. And AUG or ATG is basically start codon, and this encodes for the methionine, which initiate the polypeptide chain. While there are stop codon, and all of these generally they are represented by 2 to 6 triplets except tryptophan. In case of tryptophan, there is only one triplet. So if there is a UUA, UAG, or UGA, that all encodes for the stop codon, and they do not encode for any amino acid. So there will be no amino acid added at the end of the chain. Now, if you see this, there is a red highlighted box, UAA, UAG, and UGA. All three are stop codon. But UGG, we see in this column of the G here. So you see the UGA, UGG. So difference is in the last letter. In the first one is the A. If it's A, it's stop codon, and if it is G, then it's encoded for the tryptophan. And we have already mentioned that tryptophan is the only amino acid which is encoded only by one triplet, while other rest of them you can say it varies from 2 to 6. So minimum number of triplet encoding is 1 and maximum is up to 6. Here for leucine, for example, you can see there are 6 triplet which can represent the leucine. While for the star there is only one, that is the AUG, and it's encode methionine amino acid. Now there is a wobble hypothesis, and we can discuss it in the next, we can say, lecture.